these crabs. So here's our system of the Mesoamerican reef. I think there are a few people that are not from this region. Healthy Reefs Initiative began in 2004, and we are a collaborative effort of 73 groups that do monitoring and reporting on the health of the ecoregion. This is the groups, this is the royal we. When I talk about the initiative and all of our partners, this is who we're talking about. So it is a really large effort. Over the years, we've produced these 6 different report cards and 3 eco audits. And we have a book that talks about the kind of the fundamental concepts of reef health and social well-being. We do talk about structure and function as being important. And so this effort is a way of bringing in that functional aspect to how we can restore the health of the reefs. We start with the acknowledgement that we have a problem. So this is from our last report card 2 years ago. This was a 10-year kind of anniversary, just looking over the 10 years of monitoring that we have done. And our fleshy macroalgae had doubled in that time frame. So realizing that, we began this effort of increasing even more what we're doing to reduce macroalgae. Overall, our reef health had been increasing up until this last report, which we just issued in February, was the first time the reef health index overall has gone down. But if you look at this upper right-hand piece of the wheel is macroalgae. And this has been consistently orange, which is poor, yellow is fair, over the entire decade in a 12-year time series now. Here's just a snapshot of where we are in the Mesoamerican reef right now. Our last report card that we just launched in February included 286 sites. Coral cover is a kind of decent, it's a fair 19%, but that macroalgae is still poor at 20%. It had gone down a few percent from the last time, but we are determined to kind of get that down even further. When you look at a cross-section across the different countries, this is the one indicator that is consistently poor. Everything else it moves around a little bit, with Honduras and Guatemala having lower fish abundance, but then you have coral covers just kind of different in different countries, but this one is across the region a problem. So our idea from the beginning of our very first report card issued in 2008, we had a recommendation in there about protecting parrotfish. That seemed like the easiest approach. They were being fished, but they were not really a target fishery. They're not a high value fish in our region as they are in some parts of the Caribbean. So we started that effort, and I'm happy to say now all 4 countries have protected parrotfish. Only the coastal region of Honduras remains, but this we see as a great achievement. It is already increasing herbivory in Belize, where it's had the longest standing protection, and I'm sure this will help over more time in the rest of the area. But given the amount of nutrients we have, and that kind of goes to that bottom part, the parallel efforts to reduce nutrients, we know we need more herbivores. So the crabs and the diadema are part of that story. But just to make sure it's clear that everyone realizes, we do recognize that the pollution aspect, the nutrients that are fueling the macroalgae and also possibly putting pathogens that are hurting our corals and other marine life, that's part of our larger effort, but not what we're talking about today. We're talking about herbivory, and we're also working on these other organisms, the fish and the diadema, but we're focusing on the crabs. And where this started for me, for Smithsonian, it began in the 80s with projects, here's the picture from the 80s, where mariculture options were studied in the Eastern Caribbean, several places by Walter Adey. And this is a picture of the book over on the right here. I guess you can see my cursor, where they talk about how you can use a really low-tech process to culture these crabs, feeding them algal turfs and growing them for mariculture, for selling as seafood. So their idea was to grow them up to full scale. So that sparks an idea. Then we hear from Jason, who will talk to us next about his work. And I heard about this from Lisa Karn, Fragments of Hope in Belize. She had seen Jason's talk at a GCFI and got really excited. And so I met him at the Reef, it was the Coral Reef Initiative in Fort Lauderdale back in 2017, I think. And we cooked up lots of plans for testing what he's seen in the Florida Keys, which he'll tell you about next, in Belize and the Mar, and being able to get this really rolling as both, potentially a livelihoods, supportive livelihoods for fishermen, because they can grow these things, but also for, they could be exported, it could be sold. Here is a big Caribbean king crab going to the White House, if you can see that. That was when Reagan was president. That was when this whole thing started with you can see the little seaplane, and this was their box to put the cage that they put the juveniles in. So you'll see some of how this compares to the work that we're doing now in Mexico and Belize, but it's just a nice little throwback to where, sometimes these projects, you have ideas and they come around and this could have succeeded, but the funding ended. So they just kind of left it. And now we're picking it up again. So I'm just happy to be able to give back a shout back to that effort that started this, and we're building on that. And now the new team is taking it in the Mar, and want to recognize our funding from the Oak and Summit Foundations for the core funding, and then the Smithsonian Conservation Commons and National Geographic for this dedicated crab projects. So now I will turn it over to Jason. Let me end this. And Jason, you can push your share screen and go ahead and tell us about the history of the ecological, natural history of these crabs. Jason is a professor at the Keys College, the College of the Florida Keys, I believe. He was with Old Dominion for his PhD.
Absolutely, thank you. Is my screen showing up?
Yes.
Okay. So just kind of, thank you, Melanie. And I just wanted to kind of give a brief overview of the natural history, the ecology, and then touch on the potential role in coral reef restoration of these crabs, and specifically on some work that Mark Butler and I conducted during my dissertation, and that we've kind of expanded upon here a little bit. So in general, these large crabs come from a fairly diverse and kind of interesting family of crabs, the Mithracidae. And these photos come from a paper in 2014 when the crab that we're talking about underwent one of its first taxonomic revisions of, it seems like 10 over the last 5 or 6 years. But among the large and diverse Mithracid family of crabs, first Mithrax, then Damithrax, now Maguamithrax spinocismus is the largest and probably the most conspicuous and potentially the least abundant member of that family. So we do refer to it, and several others have, as the Caribbean king crab. However, that's a bit of a misnomer. It is not a king crab. It's actually a large spider crab. It's gone by several other common names as well. The channel clinging crab, the West Indian spider crab, but the name that, well, I was going to say that doesn't change, however it has quite a few times over the last few years, is its taxonomic designation. So right now it is Maguamithrax spinocismus. It's kind of a fun story, and I apologize to any of the authors if they're on this talk, but Maguamithrax is an awful genus. It is literally named, it is a large spider crab, and it's named for the actor who played Spider-Man, Tobey Maguire, for many years, which is just terrible. Like I said, it is the largest member of the Mithracid family, but it is also the largest crab, the largest brachyuran in the Western Atlantic, with males getting up to 3 to 5 kilograms and between 18 and 20 centimeters across their carapace width. What's more impressive, though, than its size is, I'm sorry, I jumped ahead. So I just kind of talk about the general life cycle of spider crabs, and in particular this species. Adults brood eggs in their abdomen, just like other species of crabs. And then under some stressful conditions will hatch a non-swimming pre-zoeal larval stage. Although when conditions are right, they just hatch to a swimming zoea. Goes through 2 larval stages within the first 12 to 48 hours after hatching. and then spends about 3 to 4 days as a post-larval megalopa, which then metamorphoses into a benthic juvenile within the first 4 to 7 days after hatching. So their larval development is actually very abbreviated and rapid. But it is this linkage between early metamorphosis to juveniles and growth to large adults that we know, much less about. Pardon me. There have been a number of different growth estimates or growth curves and models trying to anticipate the time it would take for these small crabs to reach what back in the 80s and the 70s, 80s, and 90s was a saleable size or a market size for these crabs. Thankfully, the target size that we identified for outplanting for restoration purposes actually falls within the size range that they're talking about here on these models. So we do not right now have good estimates or accurate models that predict with any degree of certainty how long a crab would take to get to a marketable size. But that's one of the things that we're looking into moving forward. However, we do have quite a bit of data that shows us that within the first 5 to 6 months, we're going to have crabs that are roughly the size, about 30 millimeters carapace width. These are both length, but width, that are appropriate for restoration. But their rapid growth and that abbreviated larval cycle are what make them so attractive as mariculture candidates. The other thing that makes them so attractive for mariculture is that they really taste good, and there is a pretty good following throughout the Caribbean. This is Dr. David Vaughn in Placencia, Belize, in January of 2019, where he, Mark Butler, and a couple of other folks came out and we actually tasted Caribbean king crab in a restaurant there. And I seem to remember that the cost for a plate of crab was actually more than the cost for a tail or two of lobster in the restaurant. So they are potentially of great economic value. Oh, all right. But what's more interesting about them than their mariculture potential or their potential market value is the fact that they are largely herbivorous, which again elevates their value as a mariculture candidate, but also includes kind of an ecological benefit that you can gain from this. So my former major advisor and one of his other former students recognized this and did some work quantifying their consumption rates and positing upon their potential effect on algae on coral reefs. However, one thing that sticks out is that throughout their natural range, their natural abundance or their natural density is incredibly low. So my hypothesis moving into my dissertation work was what happens if we mess with the position of the decimal point in their density. If we increase their density on reefs that are degraded and covered in macroalgae and have this algal issue, what effect does that have on the reef community? And so I went through and selected a series of small nearshore patch reefs that are demonstrably, as you can see in the photo, are absolutely algal dominant. There was very little coral cover, lots of algae, and particularly some of the chemically defended stuff like Dictyota and Halimeda that are such a problem because many herbivores avoid them. And then I applied a series of different treatment groups. In the interest of time, I won't go through them. However, I followed macroalgae cover, and this is percent, the cover in percent of macroalgae on the reef surface through time in this first set of manipulative experiments. And on reefs to which I did nothing, algae cover, as you can see, was extremely high, between 70 and 90%. However, just by adding crabs and by first removing algae and then adding crabs, we effected an enormous decrease in the cover of macroalgae through the 1-year experiment. And just by adding crabs, this equated to about an 85% reduction over the control or under the control algal cover. So with data this beautiful, especially as a graduate student, you don't trust it. So we went out and repeated the experiment about 15 kilometers away at another series of inshore patch reefs. And we added this fourth treatment group where we just removed algae and didn't add crabs, just to get that fully orthogonal design. But the point is that just by removing algae and not changing the herbivory by adding crabs on these reefs, algae cover did indeed reduce to less than 10% during the treatment application. And then through the year after that we monitored it, almost returned to the control condition. However, just by adding crabs and by adding crabs to the reefs that we scrubbed, we again saw a significant and substantial reduction in the cover of macroalgae. So we demonstrated repeatedly that crabs do, when they're at sufficient densities, have an extreme and deleterious effect on epilithic algae on these reefs. However, so yes, you add crabs, you lose algae. However, we also saw on reefs to which we added crabs a significant increase, almost a 4-fold increase in the density of juvenile corals 2 years after we added the crabs. Same kind of thing going on with not only fish abundance. Here, 1 year later, you had a 3 to 5-fold increase in the abundance of fishes, but also in the richness of fishes. So a 2 to 3 fold increase in the number of fish species that we were seeing on these reefs. So to wrap it up, we added crabs to the reef, and when you do this in multiple locations, as you'll see soon, we reduce algae cover directly due to an increase in the grazing intensity locally on those reefs. But you also have these cascading effects where you increase the abundance and richness of fishes around these reefs, and also you increase, according to our results, the density of living juvenile corals, which could be an effect on recruitment, or it could be an effect on early survival of these recruiting corals. I'd like to thank a whole bunch of people at the College of the Florida Keys that are helping me continue some of this work and some other kind of tangential projects. A bunch of folks at Mote Marine Lab who helped with some of this work, and then the group at Old Dominion University who participated in the original work. And that's all I've got.
Thank you. So now I'll share my screen. Can you see my screen?
Can.
So thank you, Jason. Hello, everybody. My name is Melina Soto, and I'm the coordinator for Mexico of the Healthy Reefs for Healthy People initiative. So as Melanie has told us before, our region is facing macroalgae proliferation, and it's threatening the health of the reef we all depend on in the region. As an initiative, we are always looking for creative, innovative solutions, and based on all the previous experience and the studies that Jason just showed us, and with our key partners, as Melanie told us, Fragments of Hope in Belize were already interested in the overlooked herbivores' importance for coral restoration, but also with the Fisheries Institute in Mexico, INAPESCA, and CONAMP, which is the Commission for National Protected Areas. We decided to test those experiments to our local conditions. So from now on, when I say we, I refer to Fragments of Hope, INAPESCA, CONAMP, and HRI. Okay, so we came up with a very simple designed project for our pilot project, where we would increase the density of the crabs, which are very low, as Jason just told us, to about 0.5 crabs per meter square. And we were trying to respect also the approximate sex ratio of 1 male per 3 females per crevasse on those patch reefs. We would also leave similar nearby patch reefs untouched, so we could use them as control. And the patch reefs will be separated between each other by at least 10 to 15 meters of open sand area, in order to try to limit the crabs' movements. So the site we chose were small patches reefs nearby Isla Mujeres in Manchones Reef in Mexico and in Silky in Belize. All those sites were included in MPAs and in the no-take zones of those MPAs in order to have a bit of control and surveillance and no fishing, right? And of course, those sites were included in an active coral restoration programs in both countries. So our first challenge was, well, the collection of the crabs themselves. In both sides, we couldn't have made it without the help of the true experts, which means the local fishermen. So we collected the desired amount of males and females, and we segged them, we measured them, etc. And then we introduced them directly in their in their reef patches, in their new home. And although we thought they would be stressed from the manipulation and the moving, as you can see, they began to work on the first minute and went directly for the good stuff. And like Jason said, they have a preference for Dictyota, as you can see. So we monitored the reef patch for about 4 to 5 months, and we adapted the AGRA methodology due to the size of our reef patches. And even though we had already seen the papers, the results that Jason has just shared us, we were pretty excited by the results. So as you can see here, on the left side are the control reef patches at the beginning of the experiment and at the end of the experiment. And on the right side are the experimental reef patches where we added the crabs. And in Mexico, we saw a significant decrease in the tough algae, which is a pretty good news because those tough algae were very high on those patch reefs, and they form sediment mats that tends to suffocate corals. So, and they decreased by about 35%. And we saw a very small decrease in fleshy macroalgae, but we also saw a pretty significant, almost 50% increase in crustal coralline algae. We observed, here is the results for Belize, and we observed basically the same tendency, although there they really got a very nice results for the fleshy macroalgae. They got a very significant decrease in fleshy macroalgae, also a decrease in tough algae, and an increase in CCA. And this last result is pretty encouraging because those algae are also promoting the settlement of coral larvae and the cementation of the reefs. So this is pretty exciting. So based on all the literature and our now local experience, we began to ask ourselves, can we reproduce them? Because as you just saw, it was pretty complicated to collect them, and there are so few in the wild that if we want to scale up that experiment in order to have significant impact in coral reef projects, we really need to have a constant production, which is the topic of the second phase of that project and the topic of the following 2 presentations that you will see just after me. And we will need to ask ourselves, how many do we need? How big do we need to grow them in order to have a significant and efficient coral reef restoration program for the Mesoamerican reef? And also, could it be profitable? Can it be a business initiative? So we also need to develop, eventually a business plan for circular blue economy for local communities. So these are like the mid to long-term goals of this project. Thank you very much. And next we will hear from our colleagues from the Fisheries Institute in Mexico, how the second phase of that interesting project that began quite small but is every time growing more and more is going. Thank you, everybody. Claudia, can you unmute yourself? Claudia, no te escuchamos.
¿Qué hace? Estoy a punto de hacerlo yo.
Can you hear me now?
Sí, ya.
Oh, okay. I'm sorry. Good afternoon. Thank you very much to attend this webinar. I'm Dr. Claudia Padilla. I am a researcher from INAPESCA, that is the National Institute of Fisheries and Aquaculture in Mexico. And currently we have a project for coral culture and reef restoration. And since 2018, we are collaborating with Healthy Reefs in this new project to use the crabs to reinforce the herbivory in the restored sites. So for this webinar, I am going to show you a short video of what we are doing. And later, Sara Gutierrez, who has been working closely with the crabs recently for this project, have a presentation with some details about it. And here, as you can see, the crabs are a good consumer of fresh algae, those algae that we don't want at the reef, and they do it efficiently for two reasons. First, the big amount of algae that they can consume, but especially because they do it selectively. Currently, we have 5 females and 1 male in this tank in our facilities, and we feed them. We mark them and measure them, and we care about them, and we check the females for the eggs. And the females are kept in this cage just before the eggs hatch, and we follow the development of the eggs, as you can see in these pictures. The movement in the video until the larvae release. And 2 months later, we have baby crabs. That is now the Sara presentation, please.
I will just jump in just to remind the participant, if you have questions, there is a tool for question, and you can also raise your hand, and we will take the questions at the end of the presentations. Thanks.
Okay, hi everybody. My name is Sara Gutierrez. I work in the National Fisheries Institute in Mexico. And I've been the one that's been mostly in charge of the project with the help of my other colleagues. We've made it since November. So basically, the generality of the project, Melina already explained of how we collected them. And we have a specific way in which we did the project, which is in semi-controlled environments. This means that we have them in tanks. So we have the adult male and females in this tank. Right now we have 3 females and 1 male. And then the juveniles, once they're big enough, we keep them in this tank. So it is I say semi-controlled environment because we do have some factors that we don't control. So we have constant water flow. So water is untreated and it's just going into the tanks as it is. We have sun exposure, as you can see, which means there's temperature fluctuations. But we do have some controlled aspects, which are the most important part for us, which are their diet. So we get to feed them a lot. There's a lot of food abundance so that all the females are happy and the male can do his job. We collect algae from the ocean and we tie it to rocks as well as fish bits. And another very important part we have control of is the eggs, the larvae, and the juveniles. So like it was explained before, we put the pregnant females in these netted boxes right before they're gonna release their eggs, and that way we are gonna make sure that as many eggs will hatch and get to become juveniles at the end. And we also kind of control the interactions between the male and the female by having them in a confined space. We hope to increase the interactions they have and the amount of times the male will fertilize the females. So what we've been keeping a very close eye on is the gestation follow-up. All their gestation periods, how many times they, ovigerous females present egg masses. And in this table, you can see each female has a different color. Whenever the color is present, it means that it had eggs. And at the beginning, we had 2 what we called acts of faith, because it was the beginning of the project. We kind of didn't really know what was going on, if it was going to work or not. But we, as Jason suggested, kept faith. And then around 31 days later, we found recruits. We found baby crabs, which was very, very exciting after a very long wait of seeing no results. So we've kept track of everything about these females that we can. They're not only about their gestation, their eating patterns, the male distribution in the tank, seeing if the male was closer to some females at specific times, so we could gather as much information as possible. And we also looked at successful batches. And what I mean with this is that seeing after the first act of faith, we were going to have a wait of around a month after they released the eggs. We expected to have the same pattern in the times that we were going to see juveniles. So in the second batch, for example, which were the orange and the green one, we expected to see juveniles around this time, February, but for some reason it was a not successful batch. So we had no juveniles come out of that batch. And at first it was very confusing, very kind of discouraging, but we gave it a thought was that temperatures dropped a lot during those days, and maybe that might have affected the development of the eggs. But even though that one was not successful, we did have other successful events, and we've had many more females coming up with eggs. And one of the points I'd like to stress in is the fact that it's very hard to see the juveniles, and that's why it's easy to lose hope. But these 2 pictures showed 2 different juveniles. This is one, and this is another one. So you can see and understand how hard it is to see them and how we kind of despaired for a little bit. But after that, we are back on track. For a while, we didn't know what really was going on, but we do have juveniles. So we also kept track of their gestation follow-up by looking at the coloration of the eggs. And you can see, depending on the day, the eggs had different colors, and that way we could keep an eye on them and know when to put them in the boxes. So you can tell that the color changes a lot from day 1 to day 31. When they look clear and pale like that one, is that they are ready to release their eggs, and that is when we put them in the boxes. We also kept track, as you saw in the video, of development of the eggs to understand a little bit more about their general development and any interesting facts. And all of these pictures show when they started developing. We saw that the eggs, the eyes were one of the first structures to start to be visible. And you can see the change in coloration in all of the eggs. And then finally, we saw juveniles. So it's been quite successful. We've had females with eggs throughout the whole time. And we've had juveniles since their eggs. These are the eggs inside the net in the top picture. And then these are the first batch of juveniles that we collected. This is the rough average size that they all had. Some were smaller, some were larger, but around 0.5 centimeter was the estimated size. And then after that, we've seen them grow. We've seen them behave in quite funny ways. They like to dress themselves with algae and hide from each other. And right now, this is the biggest male we have. He is 12 centimeters width from total width, and then 4 centimeters of his shell. So right now it's been quite successful, and that's basically it. Thank you so much. If you have any questions, don't just ask them at the end and we'll try to answer them. Thank you.
Thank you, Sara. Okay, I've got Nicole's presentation here. We've got a little slide for you to look at while I pull it up. Nicole's very handy with the mixer doing videos. So she made a video for you, and we're gonna watch that right now. Oops, wait, I need to share screen. I've got way too many panels on. Okay, you guys are seeing my screen yet?
Not yet.
No, not yet.
There is a, okay, let me stop her then. Hold on. I think I've buried the, there's the Zoom box. Okay, share screen, there we go. Host disabled screen share. Okay. Are you guys seeing my screen now?
No, not yet.
You know, I think something happened, Claudia, when I made you the host, but didn't know. It says host disabled screen sharing.
Yeah, she made herself host. I don't know how.
Okay, let me go back to that participants view. That's at the bottom here, I think.
Unknown caller.
No. Okay. Oops. Oh, I'm seeing the video, but you guys still aren't. Gosh, darn it. Okay, participants. Go over to panelists, make me the host again, maybe. I still seem to be the host. I mean, sorry. All right, so you guys can hear me, but you're still not seeing me. Claudia, I'm gonna put you back to something else other than host because I think that made the problem. Let me see, reclaim host. All right, I think this is gonna work. Now share screen. All right, are you seeing it now? Okay, now you guys are seeing the screen?
Yes.
Yes, we are.
Okay, here we go. We're gonna have Nicole's video.
Hi, everyone. My name is Nicole Craig, Belize's country coordinator for Healthy Reefs for Healthy People. I'd like to share a bit about our project entitled Enhancing Herbivory to Improve the Resilience of Belize's Reefs and Coastal Communities. The project here in Belize is at the beginning phase. Even though we got things going in February, we've gotten a lot of things done so far. We currently have 3 females and 1 large male, with hopes of getting another female soon. This project is based off Tobacco Key and falls within a no-take zone of the marine protected area. We figured this would give the project a few advantages, such as not having our crabs mistakenly caught, and they'd fall under the watchful eye of enforcement in the area. Our project in Belize would share many similarities with Mexico, but the difference in environment meant we had to be creative and have a lot of foresight. Setting up the experiment right offshore meant there were a few things we needed to seriously consider, like depth and tide, temperature, structural integrity, sedimentation and wave action, predation, boat traffic, and even human interaction. We needed to choose an appropriate depth that would allow you to still work and access the cage, but allows the cage to remain submerged despite the tide. Shallower water might have a greater temperature fluctuation as well, so a bit deeper water would help to manage this a bit. Here you can see us standing next to the cage, which rests at about waist height. Wave action, currents, water movement overall could have many effects on the experiment. Too much might rattle the enclosures the crabs are in, and that would make it hard to secure them. All that rolling around might stress the crabs. Too much wave action might also keep sediment suspended, which might impact water flow into our screened enclosures. It's important that fresh, clean water is exchanged to keep constant temperature and a nice flow of oxygen. We knew the screen cages designed to hold eggs and larvae could not be placed in the sea as is, so they were placed inside a larger box that was wrapped with 1 layer of chicken wire and then coated with paint to reduce rusting as much as possible. This extra layer of wire will ensure the crabs are contained, but that things stay out as well. An animal like an octopus, for example, would still be able to get in, but we're hoping that having this set up in an open seagrass area might discourage that. To get there, you'd have to expose yourself for quite a bit of distance, and in that moment, you also become potential prey. And of course, having the experiment set up just offshore means it would not be in the way of anyone who wanted to take a swim near the beach or be in the way of any boat traffic. The daily routine involves getting out to the cages and doing well-being checks. As I mentioned earlier, we have 3 females and 1 male, and we have to make sure that they're there every day. They eat all types of algae, but Dictyota is their favorite. They'll also eat scraps of meat for some extra protein. We've learned some techniques to secure algae to rocks to ensure the food stays nearby. This way, we're sure they're eaten and we can monitor just how much. A lot of time went into designing the cages just right, finding the easiest way to access the crabs without compromising the cage's security took a few tries. Also, our goal was to ensure the environment we created had as many things that they're used to as possible. This meant adding a lot of rocks or cinder blocks, which is important since naturally they like to hide in crevices. All the above factors were important learning lessons for us, and each setup will vary based on that area's specific conditions. Our observations are important to pass down to those who will continue this project after us, which we're hoping will be fishermen and local communities. With everything in place, we wait patiently on eggs to catapult us into the next phase. Stay tuned on our social media pages to follow this project and our progress.
Stop the credit. There we go. All right. Well, thank you, Nicole. That was great. So, oops. Okay. So now, yeah, I guess I stopped sharing screen. I'll pull something up so you have something nice to look at as we talk a little bit. I think this next phase of the project, of the webinar, we want to just let each of our panelists talk a bit about what the next phase is for them, what they see as next steps for this crab project. And we'll start with Claudia, and then we'll get to your questions from the participants in the audience.
Thank you very much, Melanie. Well, for us, this is a very excited project, and I think we have very encouraged results in a very few time, and we are very happy about it. For our vision for the future with this project, I have three main points. First is that for us as Fisheries and Aquaculture Institute, we are very interested in the project from the point of view to cultivate a species that is fishery resource with economic importance. And of course, the second one is that we want to use the crabs to reinforce the herbivory in the reef restored areas that we are working as an integral action for reef restoration, to do more than only outplant the corals. And in this point, we are thinking that it's good and a strategy to release the crabs in a single site in several times with different cohorts of crabs, mainly to ensure the crab recruitment, and also to figure out how to measure this crab recruitment as well as its effect in the restoration actions. And the third point, it will be that we have corals growing in tanks, and there is a lot of work cleaning the algae of the substrates where corals grow. Then we are very happy that the little crabs can clean those algae for us, and to have a synergistic culture that will be our biggest dream. And that is.
All right, thank you, Claudia. Jason, I think, did you want to say anything more about how you see your role, you know, as a college professor and a specialist in these crabs? Like, how could you help us in the future, or what are your plans to continue this kind of research?
Sure. So I kind of see two different directions in research informing these kinds of studies. So one would be identifying the conditions and situations that maximize the mariculture potential of the species. So what conditions limit their growth, what conditions limit their reproductive potential, how best to maximize their reproductive and successful mariculture output. And then on the other end, looking at how, just as Claudia suggested, we've been looking at how polyculture of the crabs, algae, and corals can be integrated into not just coral restoration activities on the reef, but also in a nursery setting, and even in nurseries in situ and ex situ, but also kind of, you know, continuing to explore the role of not just Magwamithrax spinocismus in mitigating algal cover on reefs, but also incorporating other species of mithracid crabs who are substantially smaller but more abundant and actually gram for gram consume more algae than the big guys. So looking at multiple consumer effects and the community of grazers that could be manipulated, rather than just focusing all of our eggs on a single basket or a couple of baskets, look at as many baskets as we can find to distribute those into.
Great. Well, that sounds wonderful. I think, you know, it is this collaboration between the conservation outreach groups. You know, the Healthy Reefs Initiative is, I think, the basket that we can all come together in, and where we can fertilize each other with ideas and knowledge, and having the researchers in with the groups that actually can get out into the field, like park managers, There is a lot of interest in doing this from other groups, even that are kind of the ecotourism type groups that have students come in and stay for a number of weeks. They could potentially be the ones tending to the crabs, you know, before they are placed back on the reef. But we need to develop, you know, the whole contingent of different levels of expertise we need, the people that are on the ground and in the water with them at all of these locations. I think there's many different ways we could organize this, you know, to try to keep it as cost effective as possible and be able to really get numbers on the reef that we will be able to see a difference. And I think, you know, exploring other species of crabs is one, but also having this potential that, you know, the fishermen could get involved in this and they could potentially help raise these crabs into an area where they're going to be, they have like a rights, when we have rights-based fisheries over certain areas, they would be able to go back to that area where they had put in the juvenile crabs and harvest potentially the males later on. Maybe we leave females on the reef, we harvest the males. There's a lot of potential for fisheries management that could use, you know, nature as a major chunk of this whole mariculture process. So I think there's a lot of excitement. We're not quite there. We'd like to see it work in the cages in Belize. And we, you know, like Nicole's video said, we're waiting on those eggs. So that's where we are now. I guess I could have shared my screen so you could see me. I would like to hear from some of the audience now. We have a number of questions. So let's just see if I can pull this up. And I know, Jason, I saw that you had answered one or two of them. We're getting them in reverse order, I guess, now. Oh no, I see them in reverse order. But a lot of these questions that I'm looking at are mostly technical questions about the crabs. So Jason, are you having a look at these? Do you want to answer some of those?
Yeah, I started typing it. Got a lot of hands, so I figured I'd wait. I'm kind of looking at it. Oh, maybe that's a great question, and we do not know the answer to it. However, a previous lab mate of mine, and basically some work coming out of the Butler lab at ODU, suggests that there is now a method for determining the age of crustaceans using their gastric mill, which is one of the only hard parts that may or may not be retained through molts in crustaceans. So it is possible that we could move forward and attempt to answer that question, but as of now, we don't know. Considering the economic importance of the king crab, are you concerned poaching might affect mariculture efforts? If so, how are you considering to prevent this? Well, it's yes. We have continually brought up that point that, you know, if we treat this as a reef restoration species, then there's very little likelihood that they're going to get any more than marginal fishing effort expended upon them. However, if you do develop this species any further as a you know, desired food item, that potentially you would be encouraging a more intensive fishery for them. However, you have a couple of things that are working really in your favor in this respect. One, they're herbivores, so they don't tend to go to traps. There has been a study done using wooden lobster traps, and the average crab took, it was something like 700 trap nights to collect a single crab. So commercial fisheries are probably out of the question. As far as, you know, how would we prevent fishery interest from getting out of hand with them? I don't know that that's within the scope of these studies, at least. And one scheme that we've been talking about for a while is the little guys are what you want out there eating algae, because they're not you know, as territorial as the larger crabs. And those large males are the ones that are really attractive as a food commodity. they're also the ones that become very territorial and start moving the reducing the density by spreading the crabs out. So if say fishermen were to take on the aquaculture, the maricultural production of them, and they were producing little crabs, putting them out on the reef to consume algae, as they grew into a crab the size that was relevant to the fishery and for food, all of a sudden, you know, you have not only reduced the algae cover on your reef, but you've also basically free-range grazed these crabs in an area that needed that ecological benefit. So you're not only producing that ecological benefit that you need, but you're also providing for that fishery resource that would otherwise be very, very few and far between.
That's great. And let me read a question that I see on the screen from Tom Moore. It's how often do they reproduce? How viable are the larvae? And how difficult are they to rear? Or how long do they need to be kept in captivity? So that one is pretty, the good answer is they seem to reproduce all year, right? Although, you know, we haven't completed the full cycle in our region, but, you know, you've found that in Florida, and that is what Walter found in the Eastern Caribbean. And the larvae are, I don't know how you answer that, but they seem to do quite well, right? Viability.
Yeah, I would say that one is definitely very context dependent. So in the early studies, you see estimates of, you know, 2 to 8% survival through that metamorphic molt between Zoea 2 and Megalopy. But then in some of the more recent studies, you see estimates of up to 98% survival when you provided appropriate substrate and things like that, and space and diet and all those kinds of things. So as the mariculture protocols and our knowledge of their biology increase, I guess the success rate of getting them into that juvenile stage is getting much more efficient. And they're obviously incredibly easy to rear.
So that question about how long they need to be kept in captivity, that one depends on, you know, what your purpose is, right? So if you want to raise it to market size and get, you know, a nice 3 kilogram, you know, crab to sell to market, that's going to take how long?
Well, we don't know. So some estimates were as little as I think 12 to 16 or 18 months. And then other estimates have been, upwards of 2 to 2.5 years. We don't know. I don't know that there are any quantitative data out there on crabs that have gotten above, say, 100 millimeters carapace width. And really, you know, if you're trying to compete with king crab or snow crab or some of these other, you know, more well-known spider crab species on the market, you would need to get up to a size in that range, that 120, 130 millimeters carapace width. So we don't know the answer to that, but it's probably somewhere between 1 year and a decade.
Yeah. But that's the benefit of, you know, putting them out smaller for this restoration purpose. They will still be eating the macroalgae, and that's a much shorter time frame. Like, depending on what size we want to put them out, if we're putting them out at 5 centimeters, that's just a few months.
Absolutely. Yeah. For restoration, you're talking about 2 to 6 months or so.
And then, you know, potentially the fishermen could go back and harvest. Some of those crabs are still going to be there. You know, we don't know what those percentages are and how well it's going to work, but you are reseeding an area, and then they could potentially, when they get to, you know, the mature size, the harvest size, then they could take some. So that's kind of the idea.
Absolutely. Here's another one. The question just above that: what other algae do the crabs eat? What is their retention rate on restored reef? And what is their survivorship through larval stage? So we got through the survivorship through larval stages. Restocked individuals, again, it was context dependent based on size. So in the Florida Keys, there was a strong inflection point in survival at about 30 millimeters carapace width, which is where we got that 2 to 6 month estimate, using that size as a target for outplanting. Less than 30 millimeters, survival was roughly 20% in a 24-hour tethering study. But greater than 30 millimeters, we had 80% survival. So size is a big factor, and we think it's related to the gape limit of whatever critter is out there eating most of them. The retention rate on restored reefs. So the only data that we have on this is from my dissertation, and that was on isolated patch reefs. So there might be some artifacts in that data. However, on those isolated patch reefs, we had crabs stocked initially at a density of about 1 per meter square of surface area. And we saw in the first few weeks, somewhere between 20 and 40% of them walked away. But after that, for another 12 months, or for another 10 months, the density stabilized. And then again, I came back to some of those reefs 18 and 20 months later, and I was finding tagged crabs on the reef that they were outplanted on more than a year and a half before that time. So they do seem to show a pretty high degree of philopatry and site attachment to rugose, shelter-rich structures. And then what other algae do the crabs eat? The short answer to that is yes. We haven't found many species of algae that they won't eat, including things like Halimeda, what was it, Stylophora, basically all of the really nasty, chemically defended and calcified algae that you would expect nothing to want to eat. These guys do. But they do seem to prefer Dictyota, particularly over just about anything else.
Here's a related question from Ilse Kofner about the potential for eating a little bit of meat. Might they also eat the coral-eating snail?
Oh yeah, I think I answered that one in the chat.
Oh, you did?
Chat thing. But no, so the answer to that was we came up with the same kind of idea a couple of years ago, thinking, great, these could be those trapezid guard crabs that you find in Passalopera in the Indo-Pacific, but in the Caribbean. And we did some very preliminary field and laboratory experiments, and there did not seem to be any interest of the crabs in corallophilia whatsoever. They didn't bother the snails didn't bother them. The consumption rates of coral did not change for corallophilia. But there has been some anecdotal evidence. I cannot remember who it was that suggested it, but they swore that they have seen Spinocismus consuming fireworms in rubble zones at night. So there is some potential to look at that.
And on the other side, like a question that could be a bit related from Valerie, that if the crabs have been used with sexual recruits, and do they graze the coral recruits? And I think the crabs are even more sensitive than other organisms that we tend to use in the aquariums to keep the algae down for the coral recruit. I don't know if Sara or Claudia, who have small sexual and fragments, microfragments in their aquarium, can answer this question also relating to feeding.
I think we have a big opportunity because the crabs are from different sizes, and I am figure that the smallest crab could be useful for sexual recruits even, and in bigger size it will be more efficient in fragments. We are trying all, and we are let you know what happens.
we have actually used another species of crabs to help us clean the algae in coral fragments. We haven't yet used them for the sexual recruits because of their size. We don't trust that they will keep off them, and they're too valuable to expose. But maybe now that we have juveniles that are so small, we could start trying to use them.
We've done a little bit of using small, not Magua mithrax, but again, the same diminutive mithracid species that that the folks in Mexico are using, Mithraculus sculptus, with some older sexual recruits at Mote Marine Lab, and it was great. It was exactly what Valerie was asking about, where the crabs are very precise in their removal of algal filaments and those fouling algae that get onto the plugs, and did not seem to have the same effect as urchins or parrotfish in or other bioeroders. So that was kind of another benefit, where they do remove algae, but they are not agents of bioerosion on the reef.
Well, I'm looking at a question from Thomas Wood about the traditional density of the crabs in these areas. From, I don't think there's a lot of data out there, right? But we do, what we know is that we don't have any records of them being highly abundant like lobsters, in the past before exploitation. Is that correct, Jason? Do you know?
Yeah, the only real estimate of their natural density that I know of is the paper from Angelo Mojica's thesis, where they estimated, I want to say that it was 1 crab per kilometer square was the average or the mean density. I don't know of any estimates from geological samples, cores or anything like that. So we don't know if they had the same kind of weird density dynamics that Diadema seem to have had, or other herbivores. So we just know that throughout their natural range right now, they're relatively very low density. And there are some areas and some different habitats that have higher densities than others. So in places like shallow hard bottom habitats, you find quite a few of them around sponges, and even in landlocked quarries in the Florida Keys, left over from mining fill for the islands and the railroad and so on. You do from time to time find very high densities of the crabs in these isolated rocky habitats with lots of shelter and productive algal communities.
And getting a little more technical, Phil Karp's the next question about, has there been any modeling of the balance and density to achieve a reduction in macroalgae that is and the amount that would be required for a commercial fishery?
No, not that I know of. We would need a growth model and some more. Probably some more general sampling of mortality rates in different habitat types. And so no, the short answer is no. We haven't looked at maintaining density through natural reproduction when arrayed against fishery mortality and repeated stocking events and things like that. No, we haven't done any modeling of that yet.
So this is all very early work. And I see, there's a question for Melina, if we want to give you a break, Jason. Melina, can you see the question at 5?
Yeah, by Iris. Hi. No, I was typing the answer actually. Well, unfortunately, we didn't see that many changes as Jason showed us, because our pilot project on the reef patch only lasted 6 months. And after the 6 months, unfortunately, we wouldn't be able to find the crabs anymore. And to answer another question I saw over there, we also went at night because also we were thinking, well, okay, we don't see them because they're hiding, but no, we couldn't find them. And then the fisherman that has been helping us went to help us also, and he wasn't able to see them either. So we don't really know what happened there, if there was some fishing or they just moved away, etc. But the main results that we saw is in that only 5 months of data were basically about the algae, because we took a lot of other data, but we didn't see much change either in the fishes or the sponges. But yeah.
Sorry, I had to mute myself. I was hoping someone else would pick a question. Interesting one from Chetwin. Sorry if I said that wrong. Does the crab help with improving the resilience of existing mangrove forests? So I don't think so, but Jason, would you offer any, have you?
I don't know.
Yeah.
I don't often find them in mangrove habitats. That's not to say that they aren't there, but it's relatively infrequent that you find them in mangrove habitat, at least in the Florida Keys. I know that there are some places out in Belize and in those out islands where there are some undercut areas in mangrove area, but that's kind of that's a very different environment than I've done quite a bit of collecting in. The vast majority of the crabs that we find are in reef habitats or hard bottom habitats, kind of rocky areas. I don't know. I don't know the answer to that. I don't know if they improve the resilience of mangrove forests.
Yes, and I don't know if anyone just saw this. Mark Butler just wrote in and kind of gave another answer about the density question. So he gave a citation to a paper. So I encourage everybody who's asking questions here. I'm sorry if we're missing them because, you know, we kind of didn't really start at the top or the bottom. We just started in on some of the questions. So I'll encourage the whole panel to read over, and anyone can just reach out with any of the questions that they feel like they have an interesting or good answer for. And we can also try to go back to these afterwards. We're recording this and it will be available somewhere. We're gonna put it, I believe, on the Healthy Reefs website if that works, or through a link to the Smithsonian space where this is being recorded. Anybody else have any others you wanna pull back up through? I'm kind of cognizant of the time. We had said this would last till about 5:15. So we can keep going if the participants want to, or we can just try to answer these on the chat box and people can read them later.
Well, there are several questions about diadema, if they are as effective as diadema, or if we have seen how do they interact with diadema, etc. So maybe, I don't know if you have more information on that, Jason?
Yeah, I just saw did you have any diadema in your study sites? Yes, we did have diadema here and there. I mean, they're almost ephemeral in most of the areas that we were working at.
We did also, yeah, but both in the control and in the experiments.
and we did not directly compare the effect of diadema to crabs, though that would be an interesting study to look at. You know, if you manipulate diadema density, do you get the same effect as if you manipulate crabs? And maybe there's an interactive effect where if you manipulate both, you get an even greater or faster effect. We don't know. but yes, there were diadema from time to time in the study sites. Not many, and in the areas that they were in, there were a lot of diadema predators that have been demonstrated to have non-consumptive effects that reduce the grazing rates of diadema. Yes, we did have them in our study sites. No, we don't know if there was any effect, or if there is any effect of manipulating the density of both of them, or if their effect is equivalent side by side.
We do have a separate pilot study underway in Honduras and Roatan that is looking at potential ways of enhancing diadema. So that we could possibly have on another webinar when he gets some results out of that study. Okay, well, I thank you all. Unless anyone has another one they want to pull out, I feel like we've exceeded the time we said we would take.
There's just a question about one that's asking if there is any evidence of the crabs eating coral. And no, there's no evidence of it. And even in the tanks we have here at the Fisheries Institute, they hate the calcareous algae. So if we will like give a mixture of algae and protein, they will almost eat everything and just leave those calcareous and algae. So yeah, no, they don't.
I've never heard of Maguamithrax spinocismus eating coral, but there is another species of mithracid that has been caught on camera consuming the apical corallites of Acropora palmata in the field. And that was one of the other species. So we've heard that story and seen some videos of that several times. But this species, no, there's no evidence that they're corallivores.
And there is a comment from Megan Davis, who has a beautiful work with Mariculture of Queen Kong, and she want to share with us the recipe to macroalgae gel diet that we are trying something like that, because when we are thinking to cultivate a lot of crabs, then the algae is the problem to get the algae. And then it will be very nice to have a recipe that you can prepare and to have available, even if you don't can go to collect from the sea. And also she's asking if we are familiar with the Caribbean king crab mariculture project in North Caicos in the late 1980s and early 1990, and I don't know if Jason know about it, because it sounds interesting.
I think that's probably what came out of Walter Ady's work, right? Because that was in Turks and Caicos, was one of his main study sites. So if I can look up the name of that facility when I go back through these comments, we can tell, but that was one of the main places where people were really interested in this. It was Turks and Caicos and the Dominican Republic. And we have that book, the Mariculture of Crab book, that describes all the work that Walter did with Smithsonian. We have it on our website, I believe. And if not, we're going to create a space where we put all of this, all of the kind of finished products. Because right now we have working space, but we'll put all the finished products in an area of our website that'll be dedicated towards the restoration of herbivory and all the different forms. anybody else on the panel found a question they want to throw up to the rest of the group and offer an answer?
Yeah, the trip was asking if we produce a how-to manual for raising the crabs, and yes, this is this is one of the products of both pilot projects that we are doing for that second phase of the project. So yeah, eventually, hopefully at the end of the year, we will be able to like produce a little manual based on what we've learned so far. So yeah, definitely, we will. Stay tuned.
Okay, well, I would thank you all for staying for the full length of this first webinar, and we will follow up, check our Facebook page, and we will be putting out the link for where this video resides. So you can look up things if you want to listen to it again or go back to a certain component. I think the chat box will either be part of that or we'll have that stored somewhere. We'll try to get back to the other questions that we may have missed. So thank you all very much.
Thank you.
Thank you.
Thank you. Thank you so much.
Okay, I'll push stop now.